In 1968, famed Japanese population geneticist Moto Kimura published a short two-page paper in the journal Nature under the unassuming title Evolutionary Rate at the Molecular Level. In this paper, he made the radical claim that the observed substitution rates in natural systems appeared to be so high that most of the mutations involved must be neutral ones. This claim and the subsequent papers that followed would make up what became known as the neutral theory of molecular evolution. In his 1983 book by the same name, Kimura defines his theory thusly. The neutral theory asserts that the great majority of evolutionary changes at the molecular level are caused not by Darwinian selection, but by random drift of selectively neutral or nearly neutral mutations. Since 1968, the neutral theory has been a lightning rod for criticism and heated discussion about the nature of biological evolution. Why was Kimura met with so much antagonism? Who were his enemies and his supporters? What evidence did Kimura have, and how has his theory stood up in the last 40 years? Buckle up, friends. We're going to dive into one of the greatest controversies in all of evolutionary biology, the fascinating world of molecular evolution. Our story begins with two giants in the field of evolutionary genetics, the statistician, eugenicist, and one of the three major founders of the modern synthesis, R.A. Fisher, as well as E.B. Ford, who was actually taught genetics by Julian Huxley, who you might know as the author of the book Evolution, the Modern Synthesis, where the modern synthesis got its name. You see, R.A. Fisher had a long-standing beef with fellow co-founder of the modern synthesis, Sewell Wright. Wright had long argued that genetic drift played an important role in evolution, but Fisher vehemently disagreed. Instead, he, Ford, and others argued that natural selection was the only evolutionary force that really mattered. He believed this so strongly that he wrote in a 1936 paper, "Evolution is progressive adaptation and consists of nothing else." However, in nature, there exists these pesky little things that Ford dubbed genetic polymorphisms. If natural selection is all powerful, why is there so much variation in nature? Shouldn't selection weed out all but the very best? To explain this, they put forward the idea that polymorphisms exist because they are either beneficial in the heterozygous state or that there is frequency-dependent selection. This means that an allele may be disfavored when it is at high frequency, but is favorable when it reaches low frequency. Thus, selection can cause the allele frequency to kind of wax and wane. To find support for their ideas, they turned to several systems, including human blood groups, as well as Ford's own study system, the Panaxia moths, which have a variety of color morphs, as I've shown here on the plates. In a series of papers, most famously their joint 1947 paper, they presented evidence that selection itself is promoting and maintaining diversity in these systems. And the latter years of the modern synthesis was dominated by what is called pan-selectionism, the idea that natural selection is all-powerful and can explain every single trait. Ernst Mayr, in his famous 1963 book, Animal Species and Evolution, claimed that even if a selectively neutral gene existed, it wouldn't remain neutral for any length of time. Kimura, in his 83 book, looking back on this, wrote that Mayr says that selective neutrality can be excluded almost automatically whenever polymorphism or character declines are found in natural populations. This pan-selectionism, as you might imagine, led to some pretty bizarre claims. The famous one is the attribution of the pink coloration of flamingos to an adaptation to blend in against the setting sun. Others have desperately tried to account for the incredible variety of shell coloration of the coquina clam, Donax variabilis, pictured here, despite the fact that they spend most of their lives buried in the sand. With the power of hindsight, the omnipotence granted natural selection seems odd, but it was likely the result of Darwinists digging in their heels against the various detractors from natural selection, chief among them being the mutationists. Nevertheless, as Dan Grauer writes in his book Molecular and Genome Evolution, a theory that explains everything explains nothing. By the mid-1960s, the cracks in pan-adaptationism were beginning to show. New methods for evaluating variation at the protein level were being developed, allowing molecular biologists to examine the levels of standing variation in natural populations across many genes, not just phenotypic traits. The biochemist John Hubby and population geneticist Dick Lewontin, pictured here, developed a technique using gel electrophoresis for sorting isozymes, which are protein polymorphisms, by their size, allowing us to visualize the amount of variation among proteins in each sample. Pictured here is one such gel electrophoresis run, where you can see the different bands represent the sizes of the protein allozymes. The amount of variation they found was absolutely stunning. 39% of the loci they examined were polymorphic. Population geneticist Brian Charlesworth, remarking on the finding, stated that, quote, "It led to an explosion of work. Everyone in the field rushed out to duplicate these studies in other organisms, including humans, and they found more and more examples of genetic diversity." In 1968, just 2 years after Lewontin and Hubby's work on natural variation, Moto Kimura published his controversial paper that fundamentally challenged the prevailing view of the modern synthesis, that natural selection was all-powerful and that all polymorphisms existed because they were maintained by natural selection. Kimura's argument was exceptionally elegant, but to understand it, we must take a slight detour and discuss the concept of substitutional load. Now, I've already mentioned 2 of the 3 founders of the modern synthesis. Here's the third, J.B.S. Haldane, seen here giving a fiery speech to a crowd. Haldane was a colorful character. He fought in World War I, was an ardent member of the Communist Party, rebuked his U.S. citizenship, and he would eventually retire in India, never returning to the United States. In 1957, he wrote a paper on what he called the cost of natural selection. This is how it works. So imagine there are 2 alleles, A and B, with frequencies P and Q. Individuals with the B alleles suffer a fitness cost S. This fitness cost, in Haldane's framework, can be thought of as genetic deaths, which would be proportional to QS. So that's the proportion that dies is QS. Therefore, the cost of selection is the ratio per generation of QS over 1 minus QS, which I've shown here at the top is the proportion that lives, and then here is the cost being the summation over all generations in, and then the cost per generation QS over 1 minus QS. Now, as an example, if P equals 0.01 and Q equals 0.99 and S is 0.1, then this proportion is 0.109, which is around 11% of the population. That means that if the population is going to maintain a constant size, then the A alleles must have around 11% more offspring in that generation to compensate for the loss of selective deaths. So how does that accumulate over time? If we set QS over 1 minus QS equal to C, and assuming that the rate of increase of the A allele is relatively slow, we can approximate the cost using just a bit of calculus, which simplifies ultimately to the negative natural log of P naught. In our example, we used P equals 0.01, so C equals 4.6. If we assume a population size of 1,000, then the total number of selective deaths is 4,600 from the time that P equals 0.01 until it has become fixed in the population. Now, Haldane pointed out that this cost places a limit both on how fast and on how many loci natural selection can simultaneously act upon. Obviously, the more loci it's acting upon, the more selective deaths must occur. Eventually, the population would just get wiped out by selection alone. So he concluded by stating that a reasonable rate of positive selection that does not result in excessive death would be around 300 generations per loci to reach fixation, and that the per generation cost could not exceed 0.1. Okay, now that we have an understanding of Haldane's cost of selection, let's return to Kimura's 1968 paper. Kimura, using empirically estimated substitution rates and the number of substitutions on average between mammals, he calculated that on average a substitution must have occurred every 2 years. He writes, quote, "This figure is in sharp contrast to Haldane's well-known estimate." He notes that under the substitutional rate that he's calculated from the empirical data, quote, "the substitutional load becomes so large that no mammalian species could tolerate it." Kimura then calculates the substitutional load, just as Haldane did, which is equation 1, shown here. Note that this S value is 2 N of E of S. That's 2 times the population size times the selection coefficient. That's what that capital S is that you're seeing in equation 1. Importantly, he notes that in the special case, where the absolute value of that capital S, again, 2 N of S, is less than 1, then equation 1, this one here, reduces to equation 1 prime down here. And note, again, that this is the load calculation. This is what Haldane calculated. And then down here is the load of that value if 2 N of E of S is less than 1. So again, when that's true, then the equation 1 reduces to 4 N of S log base E 1 over P. And the probability of fixation then is simply P plus 2N S P 1 minus P down here at the 2 prime. Again, note equation 2 is what the probability of fixation is when there is a sufficient load, when there's a sufficient cost, and then 2 prime is what it is when the cost is very, very low. Okay, so to Kimura, these two expressions overcome Haldane's substitutional load by assuming that the selection coefficient is sufficiently low that the allele is effectively neutral. This is reflected in the fact that the load of equation 1 prime is very low and that the rate of fixation in equation 2 prime is roughly equal to its initial frequency, P. Okay. In the journal Science, less than a year after Kimura's earth-shattering 1968 paper, a pair of biochemists arrived at exactly the same conclusion, heaping upon Kimura's mathematical work a litany of observations that seem to support the notion that most genetic polymorphisms were neutral. One of the most impressive evidences came from the distribution of amino acids. So they evaluated 53 mammalian proteins in which every amino acid had been sequenced and what proportion each amino acid appeared in. Next, they permuted expectations of amino acid distributions, assuming they occurred completely randomly. And then they plotted the observed against the random permutations. So the observed here is on the Y, and then the expected, assuming that their distribution was completely random, is here on the X. The fit, as you can see here, is extraordinary. So put simply, what this means is that the distribution of amino acids for a given protein in your body looks no different than if it was drawn completely randomly from a hat. King and Jukes rebut the prevailing adaptationist paradigm of the time, writing, quote, "To hold that selectively neutral isoalleles cannot occur is equivalent to maintaining that there is only one optimal form for every gene at any point in evolutionary time. We think that life is not so inflexible." Now, having traversed the history of the development of neutral theory, let's now take some time to dive into its concepts, and specifically its claims. Often today, you might hear people say that neutral theory is a null model, insinuating that it is only used as a baseline from which we can infer if selection is acting. But neutral theory makes specific predictions about what we should expect to observe in nature. So to dive into these, I'd like to tell a story that was relayed by the great evolutionary theorist John Maynard Smith. He was being interviewed by the BBC. You can actually still find these interviews on YouTube. And he tells a story about inviting Kimura to speak to his class of students. He states, quote, "The one thing I remember of the evening was the answer to a question that I asked him. I said, Look, Moto, there are really two parts to the neutral theory. The neutral theory, first of all, says that changes in molecules that occur mostly occur because they're selectively neutral. And the other one says that if you look at the standing crop of variation at any one time, most of that variation is neutral. Now, it's logically possible that one of those statements is right and the other one is wrong. How would you feel if this turned out? You know, that you're wrong about substitution, but right about polymorphism. And Kimura responded, it is logically possible, but it would be very inelegant. Zooming out, let's walk through what I think are the 5 most important posits of neutral theory. From here, we'll dive into them in a little bit more detail. So 1, most new mutations are selectively neutral. 2, most of the variation in populations is neutral. 3, that the expected levels of diversity are predictable at mutation drift equilibrium. 4, the probability and the rate of fixation is determined solely by genetic drift. And then 5, that the rate of substitution for a neutral allele equals the mutation rate. Now, as Maynard Smith pointed out, these posits are, to some extent, independent of one another. One could be right and another could be wrong. But if the base premise is true, that most evolutionary change at the molecular level is caused by genetic drift, not by natural selection, then all 5 of these are necessarily true. To get some expectation about what each of these points mean conceptually, let's dig in a little bit on the mechanism of genetic drift. From there, we'll derive an expectation for standing levels of diversity within a population, and then we'll apply the substitution mutation rate equivalency to the concept of the molecular clock. Okay, so genetic drift is the chance fluctuation in allele frequencies through time due to the fact that populations are finite in size. If you think about this statistically, it's akin to sampling error. If you toss a coin, you have a 50-50 chance of getting heads or tails. And if you could flip that coin an infinite number of times, you'd get exactly that distribution, 50-50. But if you flip it a finite number of times, you're likely to not get exactly 50-50. You might get, something very close, but it won't be exactly that. In fact, the more times you flip it, the closer and closer you'll get to that 50-50 distribution that you expect. This is more or less how drift works. When populations are small, allele frequencies diverge more generation by generation. You can think about this as you're flipping the coin fewer times. When populations are large, i.e. you're flipping it many times, they are more stable allele frequencies. However, it's important to bear in mind that genetic drift, in the absence of any other force, ensures that the life of any allele is transient. The fate of a new mutation is always either loss or fixation. They never last forever. So I've given 2 simulations shown here on the slide. In the top, this is for a population size of 100 and a starting allele frequency for the A1 allele as 0.5. And in the bottom, this is one for 1,000 individuals with the same starting frequency. And you can see that when the population size is smaller, again, I've done 5 replicate populations for each one, that allele frequency varies a lot more, right? Note that I also have an infinite population, and that's the black line. You can hardly see it, but right here at 0.5, for an infinite population, the frequencies don't change. This is basically Hardy-Weinberg, right? That there is no sampling error, and the frequency will stay exactly the same. Okay, let's think about the probability of loss versus fixation. To do so, let's take a slightly more complex case than just flipping a coin. Let's think about a 6-sided dice, where the probability of rolling a 1 is 1 out of 6. In this example, 6 is our population size. Since the probability of rolling a 1 is 1 out of 6, the probability of not rolling a 1 is then 1 minus 1 out of 6. Note that from this information, we also know that on average, it should take us 6 rolls to get to a 1. Bear this in mind. This is going to come up again later and is super important. So how does all of this relate to population genetics? Well, you remember that eugenicist asshole R.A. Fisher from the beginning, and how I told you that he had a rivalry with Sewell Wright? Well, our old friend and renowned guinea pig enthusiast, Wright applied these probabilities to the study of populations in a series of highly influential papers starting in the 1930s. So let's start by thinking about a new mutation in the population. That mutation occurs at a frequency of 1 over 2n in a diploid population. That's why it's 2n, with n being the population size. If we make some simplifying assumptions about what kind of population we're dealing with, such as non-overlapping generations, that individuals are hermaphroditic and so can mate with anyone, and importantly, there's no selection, then we can use the same math from our dice example and apply it to our hypothetical population. So, for example, we can ask, what's the probability that this new mutation goes to fixation? This is akin to asking, what's the probability I roll a 1 out of 6, with the roll representing going to fixation? Another way to think about it is, if you looked at a spectrum of new mutations, all of which start out in the population at a frequency of 1 on 2n, then approximately 1 on 2n of those mutations should go to fixation. Hence, the probability that any mutation goes to fixation by drift alone is equal to its frequency in the population. Okay, so far we've talked about mutations just kind of in passing, but we haven't really thought about their importance to neutral theory in any great detail. Now that we understand the basics of genetic drift, let's briefly introduce the concept of the distribution of fitness effects, or the DFE. One of Kimura's major claims, this is claim number 1, is that most new mutations are neutral. So plotted here is an empirical DFE that was calculated from a bacteriophage. Values are around 0 on the x-axis are neutral. Here, this is the relative fitness effects. And then anything less than 0 are deleterious. Anything above 0 are beneficial. So you can see from this DFE that the mean is slightly skewed to be deleterious, but near 0. Assuming that most DFEs in nature look like this, what are the implications for the neutral theory? If the average mutation is deleterious, wouldn't that mean that selection is acting more often to determine the fate of mutations than drift? Wouldn't that fly in the face of Kimura's initial point? Enter Tomoko Oda, Kimura's graduate student and a star population geneticist in her own right. Oda, pictured here with the entire Kimura lab from 1976, she's the one here in the center, showed that when the expression for n of e of s is less than or equal to 1, then even deleterious mutations fix at the same rate and with the same probability as neutral ones. Her nearly neutral theory is largely seen as an extension of Kimura's classic neutral theory and has received considerable support over the years. Okay, now that we've discussed the basics of genetic drift and addressed Kimura's first point, the neutrality of mutations, let's dive into his second and third claim, that most polymorphisms within a population are neutral. He and his advisor, James Crow, in 1964 derived expectations for the standing levels of diversity under genetic drift alone. So shown here is the derivation for that probability that any two individuals are identical by descent, which means homozygous at any given locus. The H sub t, this value here, is the probability that individuals are different or heterozygous. The first part here, this expression, is for mutations, the 1 minus mu squared. We square this because mutations can happen along either lineage. Then the 1 on 2n here, this is what we've already seen. We know what this value is. It's the probability that two individuals are homozygous. You can think of this simply as one person rolls a 1 from the 6-sided die. What's the probability you also roll a 1? Where rolling the same number indicates common ancestry, or in this case, that you share the exact same allele. Now, the second half of this expression is the probability that you are not homozygous, which is first 1 minus 1 on 2n, again, is like the probability you didn't roll that 1. And then we multiply that by the probability that you were already chosen to be related in the previous generation, which is 1 minus h sub t minus 1 to indicate the previous generation. Now, if you're familiar with mathematical expressions, you might recognize that this is what's called a recursion equation, where t minus 1 can be t minus any number of generations into the past. It just depends on when you started iterating. So in this way, the second chunk can be thought of as the cumulative probability that you are not identical by descent in this generation, which means that you either were in the previous or you weren't in the previous and you aren't in this one. So at equilibrium, what we can do then is set h sub t equal to t minus 1. And then we can do a little bit of algebra to derive the expected equilibrium level of genetic diversity in the absence of selection. I've shown here all of the steps, or not, I've shown here these steps to get there, but I have skipped a couple of them because what we basically are going to do here is ignore any terms that result in mu squared or mu over n. As these values are really, really small and they don't impact the diversity much, and keeping up with them makes the expression messy. So note, by the time we get here, we're approximating this. We have ignored the mu squared and mu over n terms. So those are the steps that I'm skipping. If you're worried about that or want to see the full derivation, let me know and I can show you how to do it in the comments. Thus, at mutation drift equilibrium, the standing levels of diversity should be around 4n of mu, as I've shown here. This simple expression makes a very powerful prediction that genetic diversity should scale with population size. Now, prior to the neutral theory, it was unclear if this would be the case. If natural selection was constantly acting to purge alleles or to promote polymorphisms to specific frequencies, there's no real expectation that population size and diversity should be related to each other. But neutral theory makes a testable prediction that if we look out across the tree of life, organisms with larger population sizes should have more genetic diversity than those with smaller. Now, let's move on to Kimura's fourth point, the rate of fixation. We've already noted that the probability of fixation is determined by drift is just equal to the initial frequency of the variant in question. But conditional on it fixing, how long do we expect it to take? The plot here on the left, this one here, was produced by James Crow, where the x-axis can be thought of as the selective advantage, the v sub s term, relative to drift, the 2n of e term. When this value equals 1, the allele is completely neutral, with values less than 1 being deleterious, greater than 1 being beneficial. And then the y-axis is the expected amount of time it takes to reach fixation. Note that when v sub s is 1 or less, the time to fixation, again, conditioning on that it's going to fix, is consistently around 4n generations. Now, this means that when a deleterious allele fixes, it does so in exactly the same amount of time as a neutral one. A beneficial allele, alternatively, always fixes faster than either a neutral or a deleterious one, which makes logical sense. Now, there are several ways we can show this mathematically of why this fixation value converges on 4n of e, but I think the easiest to understand is if we consider this process backwards in time, i.e. in a coalescent framework, instead of forward in time, as we have thus far been discussing. So on the right here is a coalescent tree, and you read these from the tips going backwards in time. Coalescence just means the point in time when two samples from the modern day find a common ancestor. Just like forwards in time, the rate is 1 on 2n per generation because there are 2n possible ancestors to choose from each generation. Therefore, the probability that any two samples choose the exact same one is 1 on 2n. Remember from the dice example that the expected number of rolls to get a 1 was 6. Well, for any two samples, the expected amount of time until they find a common ancestor is the inverse of 1 on 2n, just like with the dice, and so is 2n. We can expand this to any number of samples, k, which is 4n k minus 1 over k. So as k grows large, this value converges on 4n. Thus, on average, we expect a typical population pedigree to be around 4n generations long. Note the amazing correspondence here between forward in time genetics and coalescent genetics. We've reversed the time scale, but the results are still exactly the same. Now, the final critically important point that I want to discuss is that if neutral theory is correct, there should, in general, exist what has been called a molecular clock. Now, the concept was first proposed by physicist Emil Zuckerkandl, hope I'm saying that right, and Linus Pauling, but was translated into the language of population genetics by Kimura. Now, consider the following based on what we've learned so far. Let's say we want to estimate the rate of substitution between two diverged species, and we'd like this rate to reflect time so that we can use the number of differences between species to estimate the time since they've diverged. Let's call the substitution rate k, and note that it should equal the product of the probability of fixation, 1 on 2n, and that the rate at which new mutations arise in the population, which is 2n mu. Now, as you can see here, the 2n's cancel, and we're left with Kimura's famous result, k equals mu. Now, this result has some incredible implications. First, it means that if we know the mutation rate, we can use it to directly estimate the amount of time separating any two species, assuming that most of the divergence between them is neutral. But how would we know if it was neutral? So plotted here is a graph of several different genes and the amount of divergence between them across the tree of life. We've got birds, reptiles, mammals, reptiles, fish. We have lampreys, insects, et cetera, et cetera. Lots of different organisms represented in this plot. The x-axis is in millions of years since divergence based on the fossil record, and the y-axis is the number of changes in amino acid residues between any two compared species. Note that while the rate, i.e. the slope, itself differs, it's incredibly constant across species. As you can see, all of the pairs fall along the line, right? They don't jump far off of this line. Now, this constancy is exactly what we'd expect if most divergence is neutral. Okay, returning now to the 5 points of neutral theory, we have walked briefly through the mathematics and given some biological examples in support of Kimura's ideas. But neutral theory was proposed over 50 years ago. How has it fared into the genomic age? As you might imagine, given the history of its emergence, the neutral theory of molecular evolution has faced some pretty fervent criticism since its very beginning. And that criticism has not ceased. Recently, the journal Molecular Biology and Evolution ran a 50-year anniversary of Kimura's 1968 paper, which invited writers across population genetics to contribute. Andy Kern, who was then at Rutgers, but is now actually at Oregon, and Matt Hahn of Indiana Bloomington contributed a scathing piece, asserting that the neutral theory never had any explanatory power, has consistently been misused, and even at its foundations was a poor null model of evolution. So, has consensus been reached about the validity of neutral theory? Is it actually bunk and we've just been duped for 50 years? Not at all. Several of the most influential population geneticists of our time, as you can see, this paper here includes big name population geneticists all across, including Brian Charlesworth, who we mentioned before when we were talking about Hovey and Lewontin's work, have responded to Kern and Hahn's paper. And they insisted that the neutral theory was as important 50 years ago as it is now. I have screenshot here. There are 5 major points, but I'm not going to read each one of them. Feel free to pause the video and read through them yourself or download the paper that supports neutral theory. The most important point, though, is point number one. That a large fraction of the genome of organisms studied to date is subject to mutations that are effectively neutral with respect to their fitness effects, and hence evolve under genetic drift. The other points clarify and kind of expound on exceptions, but this is the bread and butter of neutral theory, that most evolutionary change at the molecular level is due to genetic drift. If that's true, everything else follows smoothly. In conclusion, there has only ever been one true challenge to the supremacy of the Darwinian mechanism of natural selection, and that's neutral theory. The incredible success of neutral theory relies on its strong theoretical underpinnings, rooted in mathematical population genetics, and its exceptional predictive power. Furthermore, it has served as a superb null model from which tests of selection at the molecular level has been derived. Neutral theory has contributed to more fields than we realize. Phylogenetics, with its reliance on substitution rates and molecular clocks, was dramatically influenced by Kimura's work. Conservation genetics, which seeks to utilize genetic polymorphism data to infer population sizes of threatened species, would just literally not exist without Kimura's insights. Without a baseline expectation of the amount of standing diversity in a population, we would be completely incapable of inferring the action of natural selection on a genome, which leaves signatures of reductions in diversity where it has targeted. But perhaps most importantly, Kimura challenged evolutionary biologists to stop telling stories. That adaptive storytelling, while appealing, was just that, stories. He challenged us to look beyond simplistic explanations, to be skeptical, and not just accept that adaptation was occurring because it made logical sense. Was it actually mathematically plausible? Is it really the best explanation for the data at hand? On a personal note, I made this YouTube channel because I saw far too often these same adaptive stories cropping up, these just so tales of adaptation. Well, it is good because it exists. Why else would it exist if it wasn't good, right? At least since 1968, we've known better. To this day, biologists are still kicking and screaming at the notion that alleles, proteins, genomes, phenotypes, or even behaviors could evolve in the absence of natural selection. As Einstein himself once complained about quantum mechanics, God does not play with dice. If there's a God at all, I can assure you, she does play dice. She's a regular statistician. I hope you enjoyed this video and that you found it informative. If you have any questions about neutral theory, please drop them in the comments. I'd absolutely love to chat with you about this more. Thanks so much for watching, and I'll see you next time.